Hi everyone. In this video, we will explore the marker-based annotation approach, and here we will be taking three different marker databases. The first, we will use our own custom canonical markers, and then we will take the CellMarker, the database, and also PanglaoDB, another databases, and then against these three marker database list, what we will take, like we will take each of our clusters and take that top few genes and compare that, and we will come up with a very simple overlap based approach. I will show you what that approach is. This is non-statistical based. Basically you are doing the Venn diagram, right? How many genes overlap, and based on that you are doing some Jaccard similarity approach, and using that we will be able to identify or we will able to annotate each these clusters. And so we will cluster against each of these, and then we will come up with a consensus, like how each of these different marker databases gives what kind of annotation. So the idea behind is to this approach is like, so that you can tomorrow you can replace this PanglaoDB with any other databases, and you will be able to repeat the same process. Okay, and you will see like how we will do the normalization of the names and how we will come up with consensus scoring, those kind of things. Okay, so that we will see. And yep, so that's what the approach is like. We will be using overlap based approach and something very simple Jaccard similarity we will use to get the top one. And we, as I said, level 3 dataset, the custom marker that you can also use your own marker list here, and the markers from the CellMarker and PanglaoDB databases. Then we will compare these three and we will calculate these metrics, like for example like how many of your database marker found in the clusters. So let's say your database has 100 marker genes for a given cell type, and your clusters, when you overlapped, out of this 100, let's say 10 overlapped, that means 10%. Or similarly the other way around, your DG top 20 markers, how many overlapped, both way, like how many of query overlapped, so query percentage and reference percentage. And based on that we can calculate very simple Jaccard index, that means how many common divided by how many unique, very simple. And so this is a very simple what is that means overlap divided by the union. So this is a very simple matrix, I would say. And if higher is the number, means that gives like the overlap is more. So again, it is no statistics, there is no confidence, but at least it will give you some metrics to compare how much overlap is there. Okay, and we'll use this for ranking. Okay, so these are the steps we will follow. We'll be doing R. We'll take these three datasets, we'll load that, we'll just clean them based on what tissue we will be doing using, and we'll process our Seurat pipeline to get the top marker genes and compare these marker genes against each of these database, and then we will score against all these three, and we will visualize, and we will do the name normalization and consensus. Finally, you will see this kind of output, like for the annotation from CellMarker, the annotation from PanglaoDB, and annotation from custom, and then we will compare them and normalize the name, and finally come to a conclusion as well, like come up this table. So this is what we will be following. So first we need to do the database. So as I said, like in the last video, the CellMarker databases, I downloaded it from the human CellMarkers. PanglaoDB somehow it's not working, but I had downloaded it earlier, so I have the data, and we are good. And canonical list, I have it, so we'll use that. Okay, and one more thing, the entire code what I will be showing, that I have will be putting in my repository, the single cell annotation course, and all the content will be there. You can later on download the code as well. Okay, so with that, let's start working. That's interesting. Okay, so the approach what we will follow is like we will three database like source, okay, canonical, PanglaoDB, and CellMarker. Okay, and we will do that. So I'm just setting my working directory, and just we need for now these 4 libraries. Okay, so my databases is there, I have stored in this path, that's what I'm loading here, and PanglaoDB data is stored here. So let me just show you this already I have covered in the CellMarker databases, like the species, the human, the what tissue it is, and what cancer type, normal, cell type, cancer type, normal tumor, and this is the main cell type, what cell type and what gene it is, marker genes. Okay, so these are the two crucial information, the cell type and the gene symbol. Okay, and this is from the CellMarker databases. And similarly, PanglaoDB also gives the similar information. Here we can filter, let's say, HS is for Homo sapiens, and then let's say we are interested in cell tissue, yeah, organ. Let's say I'm interested in blood. Okay, so and then I'm interested in what kind of cell type, right? So here you will get, erythroblast, platelets, those kind of cell types are covered over here. And we can also do multiple as well, I think so. No, okay, but yeah, so let's see this PanglaoDB, very simple databases. I have downloaded it, and these are all in the TSV file, so we will be able to load in R. Okay, so that's what we will do. So I'll go step by step. So first, this is my canonical list. This is what I am manually prepared, like, so T cell is this markers, and within T cell also we have different categories. Okay, so these are the marker genes. For B cell we have different, NK cell and myeloid, and like that, different categories are there. Okay, don't worry about this list, easily you can get it from other databases as well. But I'm just showing you, like, you can prepare a list and you can pass a list of this one, markers. Okay, so if I just show you view here, so this will look like this is a list of markers and every cell type and the what are the genes we have. Okay, so this is my first marker list, canonical marker list. Now we will prepare the PanglaoDB. So we will load this CSV file and we'll filter and we'll clean it. Okay, so I just loading the raw data, and this raw data will look like this. As I said, the same one, what species, what gene symbol, those information. Okay, I think it contains additional sensitivity and specificity, so how sensitive and how, the confidence behind the marker association with the cell type. Okay, so that's what we will be filtering, like will be, just cleaning it, like just taking the human one. So only I'm taking HS and I'm selecting these information, gene symbol, cell type, organ, specificity and sensitivity information, and wherever gene is not there we are filtering it. Okay, so after doing that, the data will mainly look like the gene, what cell type, these all are from human now, and what tissue they belong, and the confidence, specificity and specificity. Okay, so this way initially the raw data was around 8286. Okay, so now after removal we have now 7851 marker genes are there. Okay, and we can also do so just we can group by here just to see how many tissues covered, how many markers per tissue, like that. So that's what I'm doing here. I have grouped by cell type, not sorry, I'm grouping by this one and just doing the summary, and you will see that okay, so for example, Echinor cell we have around 48 marker genes are there. Okay, adipocyte progenitor 23 genes or 23 marker genes. Adipocytes we have 117 marker genes are there. Okay, so that way it gives an idea. So total we have around 178 cell types are there, and 168 plus this 10, 178, and what is the distribution of the genes. Okay, so yep, so now what we will doing is, like, so now since it contains many tissue, right, so I have written a very simple function here, like we can filter the organs. Okay, and you can pass the specificity as well. Okay, so very simple, not that difficult function. Okay, so now here I'm just doing the I'm calling this function to filter. So what I'm doing, take this my entire data and just filter with the blood and immune cell. Okay, so you can do that manually on Excel and directly, no need to do this coding, that is, but just to say that this is my clean PanglaoDB list. Okay, so these are the cell types I have in the PanglaoDB. It's around 29 cell types it covers. Okay, and again this is for blood and immune cell, because I have done the filtering, because my dataset is the PBMC tutorial what I'm using, right, the Seurat pipeline, that is from blood and immune system. So I have filtered those two cell tissue, and from that we are getting these many cell types, and just I will show you the list. So this looks like these are the B cell and these are the markers. B cell memory, these are the markers. B cell name, these are the markers. So every cell types we have the marker list, and there are 29 cell types are there for this PanglaoDB list. Okay, So now let's go to the second one. So we are now loading the CellMarker Excel file, and the raw file will, oh my god, okay, I thought I loaded this raw file will look like, this, which so again here we are selecting some important columns, and any gene is not there, removing, and comma, this we are splitting that, and nesting that, and we are removing some space, basically cleaning, not nothing much. So after this, you see around this many genes were there, but now that reduced to 53, this one, okay. And because some we have removed NA and those kind of thing, right? Okay. And so now we have the clean data set. Again, I have written the same function, and we can pass the tissue filters here. And this time we can use blood here, because blood will be there in the cell marker, right? So in PanglaoDB we had used both, because those two were the most related, but here we can give the marker. Okay. So now with this we getting around 378 cell types. For PanglaoDB it was 29, but here CellMarker we have 378. So that's what you see the granularity, right? This is more granular CellMarker database, whereas this one is a little more broader category. So that's what we'll have to sometimes we'll see similar clustering and all, same sub cell types might be annotated for a given clusters. Okay. So till the steps, what we did, we loaded 3 dataset canonical, and we loaded the PanglaoDB, and we also loaded the CellMarker. So now our data sets are ready. We are good with the next step. So now let's do our single cell analysis. This is the PBMC pipeline. I will simply run it. Okay. Mitochondria, filtering, normalization. These are standard steps. Top genes, scaling data, PCA, find the neighbors, do the clustering, and do the UMAP, TSNE visualization. And yeah, I'm just taking a backup of PBMC, and I don't want to disturb this one. That's why I'm taking it, and I'm creating another variable, and I will doing that variable every time reuse it. Okay, because once you let's say by one method you assign those level, and right, again it would be very difficult to remove those level and redo that. So that's why what I'm doing is like taking a backup of the main object, and I will be using regularly. Okay. So this is the original data I'm reviewing. That's all. So this is the original plot of the Seurat pipeline. Here only we have the 0 to 10 cluster name, but we don't know what they are. So now our next task is like we will be doing the find all markers. Okay, here we are using Wilcoxon. So we take the cluster, calculate the differential expression, and these are the markers you will see for every cluster. What are the markers we got? So these are called cluster markers, and those were marker database. Okay. So now total cluster wise, you will see that for cluster 0 around total markers, okay, 207 G markers were. These are not top, these are all the markers were there. And you can see that cluster 6 is very cluster 6, this blue one, right? It has very less marker. Okay. And very cluster number 10, you see even if 2 dots are there, very small number of cells are low, but we have a good number of markers are there in cluster number 10 here. Okay. So let's go. Okay. So now what we will do, we'll do some more filtering. That means p value should be this one, and log fold chain should be greater than 0.5, and the the gene should be in the first cell, at least it should be more than 25% of the cell it should be expressed, and for the second one specificity, like it should be less than like that. So I mean we are giving this kind of filtering. And furthermore, what I'm doing here is like this 2% right, how many expressed in first group versus all other group. Okay, so that difference is a very important parameter. So that means if it is more expressed in more cells but less expressed in other cells, not less, like less percentage. Okay, so that will give the specificity score. Okay, so and just we are multiplying that with this log fold change, that gives the rank. This is a alternate way of ranking so the genes. So the advantage, like we are in this rank score, we are not only using the log to fold change, but at the same time we are also using PCT1 and PCT2. So that means three information we are combining together to come up with a very nice score that's called rank score, and using this we will be able to find the top markers. Okay, so otherwise people generally do average log to fold change based clustering. That is also fine, but still this is much better approach, and yep. So now let's find these top markers. Okay, and just show you like how many clusters we have. 10 clusters we have, and now we got around 18,000. The dimension of the top markers is this one. Okay, so let me just show you how does it look like. So this is the cluster 0 markers. Okay, and this is what you get from the Seurat one, right? Average log to fold change, %1 and %2, and adjusted filter. And this is what we calculated on the top using these three information, combined that to get the rank score. Okay, the specificity score is nothing but the difference between these two, and the rank is this multiplied by that. Okay, so this rank score, high is the rank, means it's a good one. Okay, so that way. Okay, so now, so this shows the total. Now you see, earlier the total markers were there, but now the good markers, top markers, are actually, this is not total marker, this is the top markers. My bad. Let me just modify that. So now you see that for cluster 0 now 53 top markers seen are there, cluster 1 81, but now you see cluster 6, this group what I was showing earlier, 80 genes were there, but after applying those filters only 2 genes are there. So that's what, only 2 genes are differentiating this number of this one. So here you can further increase, 2 to 5 and 10 you can manually add it also, but when you don't get any information on the cluster 6. Okay, but that's why this number is very important distribution. You cannot directly blindly go, just see the how many top markers you are getting for each clusters. Okay, perfect. So now comes to the main part. So far we did loaded the 3 dataset, and our top markers is ready, and our reference markers, marker database is ready. So now we will do the overlapping. So here what I was doing, like, so I have created a function. What it will do, it will take every clusters and it will calculate the overlap score. Okay, and overlap score is nothing but, don't worry about the code. Okay, it is very simple one. We are calculating this output. Okay, what is the cell type and how many overlaps are there between these two. Okay, for a given cluster and for a given cell type, how many overlaps we got, and how many from this query overlapping, how many percentage of the genes overlapping from the reference. Okay, and some just statistical. Okay, so let's just run it, but you will get an idea. And this is the function we are for every cluster, taking every cluster we are doing it, right? So we have to use this for loop to do that. That's what this does it. So now let's run all three. Okay, so I will just run with the first database, that is the canonical mylist. Okay, so let's run that. And let's run the 3 database. Okay, so you will see these 3 results. This is the results from the canonical. So what it says that for this cell type, okay, and for the cluster 0, this is the canonical source. These are the markers that's matched. Okay, and like total overlap was common was 3, and reference 4 markers were there in the reference, and but out of which 3 matched, so percentage is 75. And in the query we had 15 markers. Okay, that is not that. Okay, how many percentage of query covered? 15% of the genes of the query is covered, like that. Okay, and these are the matched genes. Okay, so that way we have done it. Same if we go with the second cell marker, the results will be same for every cell marker databases, for every clusters, for every cell type, which genes are what it is matched. It will give, and that's where the Jaccard index comes into picture, which helps us to use this one to annotate it. Okay, find the top one. Okay, so first of all, don't worry about this code. Because that's where these automated tools are available, which we will see. But I wanted to show you how those tools, right, when you use it, how does it work, the behind the scene. They take this, they do that, and this all statistics they calculate and they do that. Okay, so now let's combine all the results. Okay, my bad. Okay, so now I'm combining all the results, and we can export this one into some files. Okay, so now let's just take the top three. Okay, top three. I'm just showing the top three. Okay, basically this is filtering only. Okay, so what we are seeing, like for cluster 0, okay, give me top three from each database. Okay, so as per cluster 0, we were getting CD4 T type and T cell, and from as per CellMarker we are getting this name. Okay, central memory CD4 plus T cell, CD8 plus, and names this one. Okay, and as per PanglaoDB we got these three cell type. Now you see every database is giving different nomenclature. Okay, so that's what we need to normalize the name. Okay, and it would be very difficult. Okay, but now what we will do, instead of see 333, what we will do, we'll take the first top one. Okay, top one hit, I'm just doing it. Okay, this is the top one. So now you see for cluster 0, what canonical told, it is this one, and what CellMarker is saying, what PanglaoDB is saying. Now for cluster 1, T cell, T cell, T cell. Cluster 2, B cell, B cell, B cell. Cluster 3, again T cell, T cell, but here you see some NK cell, some little bit mismatch is there. Okay, so that way, what each of the sources is telling, you can sort that, take the top one, what we have taken it. Okay, so now let's do the visualization. Okay, and so now we have the top 11 clusters we have, right? 10 plus 1 starting from 0, and we will just visualize it. Okay, so now this plot is from as per the what the first canonical marker. So as per canonical marker, this is like CD4 plus, just show you over here, and this name you see, right? CD4 plus. Cluster number 1, cluster number 1 is this one, T cell. Cluster number 2 is which one is cluster number 2? Cluster number 2 is this one, right? And 2 H per it one is B underscore cell. So you see B underscore cell. So like that we are annotating that. Okay, and so now as per the second CellMarker, you will get a different so you get a different this one, the nomenclature. Okay, and so here you see it's a mega karyote. Earlier it was I think the platelet for cluster 6. Here we are getting mega karyote as per the CellMarker database, and as per our, it was platelet. Okay, so now the third one, let's see what is PanglaoDB saying. So here it is also saying mega karyotes. Okay, so now you see we have this information, the final output. We need to make the consensus one. Okay, so now we can use something called normalization. So basically this is nothing but you can use something like, if T cell is there, you just change it to T cell. If T cells is there, that also you change to T cell. You can come up with this name and you can manually do that. It's very tough. That's where AI will help you here, LLM. But this kind of mapping is there. Okay, and I had some two different, I was just, while doing the experiment, I was initially trying this kind of this thing, but this gave me a very bad result, so I had to improve further. Then little bit better I got in a consensus. So this is what I'm normalizing it. uh, so the normalization name is now like, see, like this. So CD4 underscore T was the rate. I changed it to this name, CD4T. Okay, so like that, this is the normalized name for each of these original name, and then the main things like you join them. So basically what I'm doing here, instead of this long format, you now do the wide format. Okay, so let's do that wide format. Okay, so you see now this is the final output. So as the cluster 0, as per canonical, this was this one, CellMarker and T memory cells, and what is the Jaccard for everything. But right now we don't need this Jaccard because already we have filtered it. Okay, so this is the final output you get, and you can take this information and further, as I said, you can go to other databases, that what I have shown in the previous video, and you can do it. Furthermore, there is one more thing called, and this is a little complex in terms of this thing. You can do a voting, how many matched kind of thing, and you can calculate number of agreed, how many matching are there, like that. But I don't see any matching, so I will for this I think it will come very bad. So let me try that. Okay, so yes, same name, and we are seeing, see, everything is very conflict, and here are the medium conflict and medium conflict. So sometimes it is very difficult, but we need to use our expert judgment to come to output. Okay, so that shows like how you can manually do it. Okay, uh, by simple overlap approach. Don't worry about the code. Again, I'm repeating. Nowadays coding is very easy. Okay, with the help of AI you can take chat gpt help, but main is like understanding the concept, like how the tool is working. So that's what the main reason of putting this complex thing in this video, so that you know how the tools behind the scene works actually. So they take this one tool and they compare it, and there will be multiple iteration they do refine it, because so many very close names we will get, right? So now, as I said, PanglaoDB 24 cell types, but CellMarker 2's 378 cell types are there, right? So it's very challenging to come to a conclusion. Okay, so yep, I think that's all here I had to show you. Um, so yep, so the final output, like all are very similar, because this is a very good data set for demo, but when you will work with your real data set, you might face challenges. So that's where the trick is, like you take this one, run through one, okay, and get a plot like this, okay, and then second plot like this, and then manually check, look this, okay, very close, and take the help of that ontology tree what I show, right? In I think Azimuth, right? Azimuth single cell, if you go, so there they have given this one, right? I forgot that link, so that's why I'm referring this one. Yeah, if I go over here, so this is the ontology, right? So you can come over here, and you can search this name, for example. Okay, okay, so you can search, probably it will match somewhere, and it might give some information, and it will somewhere it will match. Okay, and there you can do the hierarchy as well. You will see this will very nicely, it will go so as the tree. Yeah, let it load. So just remember, this is ontob.org. Okay, this helps to do the ontology. I think searching will take some time here. Let's do an example, one thing, like, so monocyte only, if you select, maybe the searching will come here, and we will get this nomenclature. So you know that, okay, this is part of this monocyte group. So that way you can categorize that using this ontology tree. Okay, so with that, I think we are good, and we will move to the next tool, which is this one, the Clustermole. So this is the R package, which actually, interestingly, it queries against 7 databases. So the 7 data like Arch, CellMarker is there already, what we have taken. PanglaoDB is there, and it also compares MSigDB, Savan, Tissue, Excels, and it uses something called hypergeometric test, and using that it tries to annotate. Okay, so yep, so we will cover this one into the next video, and that is very easy to do, because not that much complexity. Yeah, I hope this was useful. It gave you some idea about how manual annotation is done. uh, yeah, so that's all, I think. Okay, okay, so I'll see you in the next video. Thanks for watching. Thank you so much. Bye.